Hello everyone, I'm Kavita from first year MSc Botany. Here I am going to present my e-content regarding general characters, occurrence, distribution, mode of nutrition and reproduction of deutromycetes-Alternaria. First, we are going to see about systematic position. It belongs to kingdom Mycota, division Eumycota, subdivision Deutromycotina, class Moniliales, order Hyphomycetes, family Dematiaceae, genus Alternaria. Some of the general characters of genus Alternaria. Deutromycotina includes those fungi which reproduce only by asexual method, producing asexual spores called conidia, and the state is called anamorph state. That is why Deutromycotina is called fungi imperfecti. It is a fungus causing some of the diseases like leaf blight on over 380 host species and other diseases of the plant. Species of this genus are cosmopolitan and can survive as saprophytic and also endophytic as well as weak parasites. It is an opportunistic pathogen on numerous hosts, causing leaf spots, rots and blights on many plant parts. It can also cause upper respiratory tract infection and asthma in human with compromised immunity. In the multicellular pigmented spores are produced in chains or in branching fashions. The spores are broadest near the base and taper gradually to an elongated beak. Formation of polymorphous conidia, either singly or in short or longer chain, and provided with cross, longitudinal as well as oblique septa and having longer or short beaks. The host range of this Alternaria species. Most of these species, they derive energy as a result of cellulolytic activity. However, species of this fungal genus are often prolific producers of a variety of toxic compounds. The effect of these compounds have on animal and plant health are not well known. Many species of Alternaria modified their secondary metabolites by sulfoconjugation. However, the role of this process is not yet understood. Some species or plant pathogens cause range of economically important diseases like stem cancer. Latent infections can occur and result in post-harvest disease or damping off in case of infected seed. Two major features of Alternaria species are the production of melanin, especially in the spores, and host-specific toxins in case of pathogenic species. They produce two types of toxins like phytotoxin and mycotoxin as secondary metabolites. Phytotoxin causes pathogenesis in plants, whereas mycotoxin causes pathogenesis in humans and animals. They are also considered as necrotrophic fungi because they invade cell nutrients of plants, causing necrosis in the plant cells. One of the species, Alternaria alternata, is a predominant fungus in this genus that grow in various substrates like food, cloth, goods, etc. Alternaria species produce phytotoxins like alternariols, altinuin, tentoxin, and tenoazonic acid. Some strains of genus Alternaria live as endophytes in crops like leaf foliage, seeds, and fruits. They are significant phytopathogens causing pre-post harvest losses, deteriorate crops quality and productivity. Alternaria alternata and Alternaria infectoria produce DHN, that is dihydroxynaphthalene melanin pigment, play a role in imparting pathogenicity. Oxidative polymerization of phenolic precursors result in production of DHN melanin that deposits in conidia cell wall. There are at least 20% of agricultural spoilage is caused by Alternaria species, with the most severe losses reaching 80% of the yield. Next, occurrence and distribution. The genus Alternaria has nearly 299 species. They are ubiquitous and cosmopolitan, strong saprobic and weak parasitic in the environment, and normal agents of decay and decomposition. The spores are airborne and found in the soil and water, as well as indoors and on objects. They can grow thick colonies, which are usually green, black or grey. Alternaria species are required high moisture conditions to colonize the kernels, such as grains, before harvest, fruits and also the vegetables. The Alternaria toxins can be found in grains that are dried in field or delayed harvest. Alternaria mold toxins are found in heavily weathered sorghum. Post-harvest occurrence of Alternaria in fruits and vegetables are common due to high moisture content along the whole value chain: transportation, intermediate storage and processing to final storage. Some of the parasitic species cause hay fever, and few saprobic species or common contaminants also occur on culture media and dead part plant. These organisms withstand low temperature and responsible for rot of several products during storage in refrigerators. Some of the common species of this genus are Alternaria solanae that causes early blight of potato. Next, Alternaria tenuis causing black spot disease on wheat. Alternaria brassicae and Alternaria brassicola both cause leaf spot of crucifers, that is cabbage, mustard, cauliflower, etc. Alternaria solanae on potato leaflet produce spots which give a blighted appearance. Spots are small, isolated, scattered, pale brown to dark colored and are angular or oval in shape. There is narrow chlorotic zone which gradually invade the normal green area of the leaflets. Next, the mode of nutrition. Alternaria species exist as saprophytes, endophytes and pathogens or parasites. Saprophytes, that where the organisms live in a dead decaying organic matter and they obtain nutrients from the dead organic matter. Endophytes, it can be inhabited in different part of the crops including foliage, seeds and fruits. The last one is pathogens or parasites. It is opportunistic pathogens that cause infections in humans and plants. A large number act as decomposers of leaf litter. And this is the structure of Alternaria. Mycelium consists of hyphae, which are light brown colored at early stage, but later on they become dark brown, slender, septate, profusely branched. At early stages of the growth, the hyphae are intercellular, later on become intracellular within the host cell. And their conidia are singly or groups form 4 different structures. These are Cinema, Sporodochium, Acervulus, Pionium. Next, the mode of reproduction. Asexual reproduction is the only way of reproduction in deutromycetes, which occurs through asexual spores called the conidia. Sexual reproduction is absent in the deutromycetes. Alternaria reproduces only by conidia, which are produced at the tips of conidiophores. The endophytic mycelium grows out as erect and aerial hyphae through the stomata or ruptured epidermis of the infected host tissue. The conidiophores are short, dark-colored, aerial, septate structures and cannot be easily distinguished from the somatic hyphae. The conidia are produced singly or in chains at the tips of branched or unbranched conidiopore. The conidiopores are undifferentiated and and emerge through the stomata of the infected leaf as we seen above. The dark colored conidia are multicellular and ooclavate, obovoid and muriform in the shape. They are both transversely and longitudinally septate. There are about 5 to 10 septa in each conidium, and number of septa is probably dependent on environmental conditions. A conidium develops as an apical bud from the uppermost cells of the conidiospore. The young conidium first divides by transverse septa and some of its cells divide by longitudinal septa. Conidia with transverse and longitudinal septa are called neuriform or dictyospores. At the time of development, a branching of conidial chain is also observed. Any lower seal of conidium produces a bud which develops into a conidium. The conidium measures 30 to 130 μm in breadth. It is surrounded by a two-layered wall where the outer layer is pigmented. A conidium develops as an apical bud from the uppermost cells of the conidiospore. The young conidium first divides by transverse septa and some of the cells divide by longitudinal septa, as we seen above. Once the conidia are divided by the septa, it gets matured and it gets detached from the conidiospore and are disseminated by the wind. In the presence of moisture, they germinate quickly by sending 5 to 10 tubes. This is the picture of stages in the development of mycelium, conidiospores and conidia of the species Alternaria alternate, Alternaria solani, Alternaria brassicae and Alternaria brassicola. And next we are going to see about the germination of conidia. The conidia, as we have seen above, it is disseminated by the wind on being detached and they germinate on suitable substratum. Humid conditions always favor the germination. Enter through the stomata of the hoist leaf and soon develop into new mycelia within intracellular spaces. Alternaric acid has been isolated from the culture filtrate of this fungus, and it is regarded that the pathogenicity of the fungus is due to the presence of this acid. Next, pathogenesis in the plant host. The spores produced by Alternaria starts the germinating by form of short germ tube. The germ tube extends form an erect and short conidiospore with enough moisture and temperature between 31 to 35 degrees Celsius. Mitosis division occur in conidiospores, develop reproductive structure conidia with short beak. Conidia penetrate plant cells and invade nutrients, causing necrosis and chlorosis. The spore of this fungi affect fruit and lenticel. Infections are generally characterized by the disease like lesions, spots, blisters and rotting of a leaf. The perfect stage of Alternaria, according to Webster in the year of 1980, the perfect stage is loculoascomycetes fungus, Pleurospora infectoria and Alternaria infectoria. Thank you.